stuff that I cover in lecture is fair game for exams. So any other exam questions while we're at it? Yeah?
Will it be hard?
I don't know. I write them. So what do you think?
There were just a lot of numbers. That's why I was asking.
A lot of numbers. Bunches and bunches of numbers. I'll probably try not to have too many numbers. Remember, the clicker question was, most in a particular country, that kind of thing. That it's 10,328 or 10,329 that I don't expect you to remember. So those of you who may be wondering why I have this ridiculous grin on my face and haven't been in my classes in the last 2 days, we just heard that NASA is going to be funding our research for the next 3 years to the tune of about $500,000. So that's why I'm ridiculously happy, and hopefully that will be reflected on the exam. We'll see, with any luck. Okay, so again, quick follow-up from last time. Filoviruses, they're basically just molecularly big, long rhabdoviruses, molecularly relatively boring, but they have high incidence of disease. And people were actually asking me last time about MSF, Médecins Sans Frontières, so I brought this piece here. And this is particular PPE that's appropriate for dealing with some of these hemorrhagic fevers. And that was really probably the big difference, which allowed them to take care of the disease, was actually getting proper PPE. And then on the back here, maybe it's a little hard to see, but here's one of the recovered patients who's now went back and was treating some of the people during the epidemic. So I'll just leave that up here if you want to come and take a look at that. So most people, yes, they're really scared about this. They make big brochures that have red alert signs on them. They don't do that for flu, which is, I think, rather unfortunate. Yeah?
Since she recovered, is she immune to Ebola? Or?
Yeah, so you seem to be, once you've recovered, you seem to be immune then. And that's partly why vaccines seem to work, because that's the whole point of that process, is if you have recovered, you do seem to have antibodies which are protective antibodies. Now, exactly what those antibodies are and how they're protective, that's a very active area of research. Okay, so orthomyxoviruses. Who remembers the name orthomyxoviruses? Paramyxoviruses, orthomyxoviruses, the real ones, the kind of not real ones. The main thing here, of course, is it's about flu. And in fact, these are the only orthomyxoviruses, are the influenza viruses. In terms of the molecular aspects of them, that's of course what we really care about, right? Yes? Is that they're segmented negative strand RNA viruses. So this is the first of the negative strand segmented viruses. What segmented RNA viruses did we already talk about? Tobacco mosaic virus is close. That's just one RNA.
Was the cucumber?
Cucumber mosaic virus. So a lot of these plant viruses have multiple segments. Thinking of things that might be on exam questions. Those are the kinds of things that I might be thinking about. So again, it's about flu, and then I will spend some time talking about vaccines because they're near and dear to me, my family, etc. And the flu vaccines, there's also some really cool technology based on understanding the molecular aspects of how these viruses work, and then being able to use those to make, I think, some really fascinating vaccines. So a couple of key things regarding orthomyxoviruses, flu viruses. Again, these are really good things to look at when you're studying for exams. Segmented genome, and then we've spent a long time talking about hemagglutinin, but bears mentioning again here, this is really that receptor binding and fusion protein, really the paradigm for the so-called class 1 fusion proteins. Neuraminidase we haven't talked about too much so far. This is an enzyme which is also one of the envelope proteins, but it chops off the receptor. And that's probably very important for having the virions be released from infected cells. And that, it turns out to be the target of some of the best anti-flu drugs, is this neuraminidase. Another previous target, but unfortunately is now almost all the circulating flu viruses have a mutation that it's resistant now to this particular drug, is the matrix protein. And that matrix protein also can serve as an ion channel. And the whole conformational change that happens in hemagglutinin, has to do with pH changes as you're coming into the endosome. Another couple of fun things about influenza viruses is that they, despite the fact that they're RNA viruses, replicate in the nucleus, which is really kind of bizarre because they've got their own RNA-dependent RNA polymerases. Why the heck do they have to go to the nucleus to replicate? Well, it turns out that these viruses don't have cap synthesis machinery for their messenger RNAs, so they steal caps from other messenger RNAs. And they do that, in fact, in the nucleus while they're being made. And so the reason that these viruses replicate in the nucleus has to do with that's where the caps are. That's where you can snatch a bunch of the caps here. So again, we'll talk a little bit about origins, but mostly about disease at the end here, rather like what we did with the filoviruses last time. The structure of these particular virions is kind of boring at the overall virion stage, but the nucleocapsids themselves are really fascinating. It's a classic envelope virus, and this is one of my favorite actual micrographs of pretty much any virion, because you can actually see these individual nucleocapsids here inside the virion. This is an absolutely amazing electron micrograph here. But the virion itself is kind of blobby, and that's really quite true for most of these. And what that means is also pleomorphic, i.e. you can have lots of different shapes, each of which then is still containing a virion. Again, binding and entry, most of the story there is the hemagglutination. Messenger RNA production, that's this whole cap snatching business. And then we have the same problem that we talked about with the philo and the rhabdo and the paramyxoviruses, which is how do you switch between genome replication and messenger RNA? How did that work with those other viruses? Presence of the NP or N protein. I'll give you one guess how it works with influenza. Presence of that nucleocapsid protein. So turns out to be extremely similar. And then getting out, and disease should probably also be vaccination here as well. So what are the orthomyxoviruses? Flu. Actually, there are 4 classes of these flu viruses, A, B, and C. Pardon me. I don't need to have little yellow lines here, although we could do that. Be interesting, potentially. A, B, and C. A and B are the 2 that really cause most human disease, and also most of the disease in some of the animals which influenza is infecting. C is a similar virus, but doesn't seem to cause much in the way of disease. And there's a D, which causes some disease in livestock. A couple of important things, and maybe that yellow line was a good thing here. It's not what's caused by a rhinovirus and a flu virus. Coronaviruses. Common cold. A lot of people say, oh, I got the flu. If you're saying you have the flu, you probably don't have it, because you should be lying flat in bed if you have the flu. So, and not be here because you would be infecting everyone else. Although all of us are vaccinated, so we should be fine. Right?
Right.
We'll get back to that a little later on. So what does it look like? Again, there was that absolutely beautiful EM that I showed you just a couple of minutes ago. This is another one of these EMs. Each of these really long elongated particles here are virions that are coming out of an infected cell. So highly pleomorphic in terms of the virions. So it's really hard if you are trying to use electron microscopy to actually detect and figure out what infection is going on there. These virions are pretty nondescript. The budding process and how these virions are produced is at the plasma membrane. So they're coming out from the plasma membrane and then being released from there. Some of the flu, I should say the orthomyxoviruses, have 6 nucleocapsids. A and B, which are the ones that we really care about, have 8. And those are the ones which are packaged inside this envelope. And here, most people would actually call this a filamentous kind of virion. But again, it really varies completely from preparation to preparation. There doesn't seem to be any real control over that particular structure, which if you think about it, kind of makes sense. There are these separate nucleocapsids that could all line up one behind each other, or all sort of packed around each other. There doesn't seem to be a real limitation in terms of what those look like. In a perfect world, it would look like this, but of course it doesn't. But it's easier to think about in terms of how these virions are put together. So nice spherical virion, never see these things. With membrane proteins on the outside, we've got our hemagglutinin here, trimeric, again, fusion peptide hidden on the inside of it. Also the neuraminidase, which is a protein that will cut off the receptor molecule from the surface, presumably of cells that are producing these virions. And then another one that we haven't really talked about too much in any other viruses, because many of them don't have it, is this, they also call it matrix protein, but it's an ion channel that goes through the virion envelope. And then there's a much more classic matrix protein, M1, bridging between the envelope proteins and the nucleocapsid proteins on the inside. 8 nucleocapsids, and we'll see what they're coding for in just a second here. Helical, but also with proteins bound to the end. What would those proteins likely to be? RNA polymerase, because these are negative strand RNA viruses. So they have to be bringing along their own RNA-dependent RNA polymerase. Complex, unfortunately named PA, PB1, and PB2. Again, historically, for all of these things. And then, curiously enough, even though, again, structural proteins is what ends up in the virion, there's also a non-structural protein, NS2, which ends up in the virion. And the reason that it was always called a non-structural protein is that that protein is present at much, much, much lower levels than all the other proteins which are present there. And we'll see what those proteins do right after I answer Ian's question.
Do we not consider that a scaffolding protein because it's in the virion?
So yeah, so if you have a protein which is present in a mature virion outside the cell, that's not a scaffolding protein. A scaffolding protein is gone in the mature virion. So it's important for putting it together, but then not for the rest of it.
Is this one not involved in, or not important in putting it together?
So we'll see what it's important for in just a second. Has to do with the host response to that. So important in a kind of similar way. You have to have your own RNA-dependent RNA polymerase, but also you need to shut down some of the cellular machinery. And that's actually what NS1 and NS2 are doing. So important ones here. Again, we've circled them. These are the ones we're going to be talking most about. HA, hemagglutinin, glycoprotein, binds to receptor, and membrane fusion. PB2 and PB1 we'll get back to in a lot more detail later on. These are RNA polymerase activities, but also involved in stealing messenger RNA caps. And so we'll talk about those in much more detail later on. And then this M2 protein, this is the ion channel, which again was the target of many that used to work really, really well, anti-flu drugs. But unfortunately, all the circulating strains now have mutations in that gene that are no longer susceptible to those drugs. So the HA protein, we've looked at this one now a number of different times. It's, again, the paragon for the class 1 fusion protein. It's made as a single protein, and that single protein gets cleaved right here with my wonderful red pen, and that generates two separate subunits, and this fusion peptide, which is the highly hydrophobic alpha helix, that when this part of the protein gets out of the way, once it's interacted with this receptor, and you have a change in pH, then this can fuse with the membrane. This fusion peptide starts down here, but after you have a change in pH, where does the pH change? In the endosome. Then this helix extends, puts the fusion peptide out here. Now it can fuse with the membrane of the host and release the genome, which is on the inside. This is a nice cartoon from the other textbook that we used many, many years ago. Is there a copy of this on reserve if people looked for it?
There's no copies.
There's not. OK. I will try and make sure we get some more over there. There's this section on flu is actually really well described there, and I think not as described as well in our textbook. So I'll try and get those over there. So when you have receptor binding, that will be here. Here we have our virus envelope. Here's the plasma membrane. You have receptor binding, goes into an endosome. In the endosome, the pH changes. That allows this fusion peptide from down here to flip up and insert into the host membrane. And as the virus is moving through here, that change leads to the conformational change that pulls the two membranes together, and also a acidification that happens on the inside of the virion. So the acidification that happens on the inside of the virion, that's due to this M2 ion channel. And that acidification is absolutely critical for releasing the genome. And so that's why it was a good target for amantadine, which, again, used to be a really nice drug against influenza. But almost all of the ion channels when in circulating influenza viruses are resistant to it now. So this is that hemagglutinin, again, one of the best studied of these class 1 fusion proteins. Starts out being made as one single protein. Then we have proteolysis. This proteolysis happens through a cellular protease, not a viral encoded protease. I didn't tell you about any proteases in that list before when we looked at the table. That's because there aren't any. And this protease will cut the HA protein as it's translated, as it's being processed through the ER and the Golgi. Once you have this cleavage, that gives you your fusion peptide, but also allows the receptor binding part here to be able to move out of the way when you have a low pH. Yeah, you have a question? I saw your hand coming.
Before I had it out, I know we've talked about this before, so forgive me if I'm not remembering, but is there a lot of, or is there any speculation about evolutionary relatedness in terms of fusion peptides? Because they all seem to have a really similar kind of structure and, you know, how they're uncoded and all that.
Yeah. So so the question is basically, are there evolutionary relationships, clear evolutionary relationships between different fusion proteins, and we'll just say fusion proteins in general? And the answer to that is it's not obvious that there is this conservation. If you just look at the fusion peptides themselves, they have hydrophobic amino acids. And they're not always necessarily the same kind of hydrophobic amino acids. But the fact that you have this cleavage followed by structural change would make one think, there might be some kind of evolutionary relationship there. But there's no obvious sequence similarity in any of these things. There's some structural similarity, and those of you who were at David Wiesler's talk I may or may not remember that respiratory syncytial virus has an extremely similar three-dimensional structure of its fusion protein to the coronavirus fusion protein. And he thinks that's evidence of some kind of evolutionary conservation. May or may not be true. It's really hard to tell with these things. But yes, there's definitely, in terms of thinking about it, highly similar kinds of processes between all of these things. But the main thing is how you get that change in structure. Here it's very much pH dependent. In many of these other ones, it's not pH dependent, it's receptor binding dependent. So there are different ways that you can get this to actually happen. Okay, so once you have membrane fusion that takes place, nucleocapsids are released. What do these nucleocapsids look like? They've got negative strand RNA, surprise, surprise, bound by this NP protein. And NP, nucleocapsid protein, again, what does it do? It binds to the nucleocapsid. But what's interesting is even though these are negative strand, and in theory, you could just have one long strand, and that's how all of the other ones, the paramyxos and rhabdos, the filoviruses, also be packaged in one long single helix. These are actually wrapped up in kind of a double helix of proteins. The nucleic acids are not base paired to each other, but the proteins seem to line up as a helix. And that gives you a loop because the 3' ends and the 5' ends are right next to each other of each of these individual segments. And these are some real electron micrographs of some of those loops, which just blows me away that you can see these, basically kind of individual chromosomes. I like to think of these as kind of viral chromosomes, the 8 segments that are present in these. Yeah?
And they're each attached to their own RNA polymerase?
So each of the nucleocapsids has their own trimeric RNA-dependent RNA polymerase complex associated with it. And that RNA-dependent RNA polymerase complex is associated with both the 5' and 3' ends. And that's probably how you get this structure, is the polymerase binds to those 2 ends, and then you end up with the rest of the helical structure. There is base pairing that happens at this very ends of the genome, but these parts are not base paired. Yeah?
Quick clarification. Are these all subunits of one polymerase, or are these 3 different polymerases?
So only one of them seems to have actual RNA-dependent RNA polymerase activity, but the other ones are probably involved in binding to the ends and definitely involved in stealing caps. So there's only one that's been clearly shown to have RNA-dependent RNA polymerase activity. And not surprisingly, it has a really similar structure to the other RNA-dependent RNA polymerases that we've talked about so far. How many nucleocapsids are actually released when you have fusion? What do people think? All of them are released because you need all of those that are in the virion. But once they're released, they don't replicate in the cytoplasm because that's where you have the fusion. So the next thing that has to happen is each of these nucleocapsids has to be transported into the nucleus. And so this process of nuclear transport for all of these nucleocapsids, various different proteins, etc., is really pretty amazing for these orthomyxoviruses. So you have the whole nucleocapsid complex that gets transported into the nucleus. These things are pretty big. So that process is still not entirely clear. They've got nuclear localization signals on these proteins, which allow them then to come into the nucleus. Yeah, Patty.
So it looks like in those images that the nucleic acid is specifically associated with PB1, and then PBA or PA and PB2 are associated with PB1 as well. Is that the molecular understanding?
I didn't add the latest, I think it's an X-ray image of the influenza polymerase attached to the RNA. I think it's just the PA which is clearly associated, and PB is probably the polymerase, PB1, excuse me, and PB2. But the details there are a little unclear exactly on how this is working. So I would just say, as far as we're thinking about it, i.e. for next Wednesday, that all 3 of them then are associated with 5' and 3' ends, and that's where they sit. So each of the nucleocapsids will have the trimeric polymerase complex that comes with it. So that gets imported into the nucleus, again, through these nuclear localization signals. What you have is now transcription or production of messenger RNA with caps. These then, messenger RNAs get exported into the cytoplasm where they get translated. Then all of these proteins have to be imported back into the nucleus where they can associate with the first anti-genome RNA and then genomic RNA, which has been made through the RNA polymerases. These then get put together. The nucleocapsids get exported together with M1 and NS2, and then go out to the Golgi where they pick up these envelope proteins, et cetera. So the amount of nuclear import and export here is really amazing, and evidently stealing caps, because that's the reason that they go into the nucleus, has got to be evolutionarily pretty important, because otherwise you probably wouldn't have all of this extra transport stuff going on. So more questions on this. And he's asked that, what does he mean? It's about to have a clicker question, so time to get your clickers out. Hopefully, again, let's see if we can do 100% for everything today. How many genome segments does Influenza A have? 1, 2, 4, 6, or 8? Again, he loves these questions because it's really easy to come up with alternative answers.
I watched this interesting video on influenza diffusion, and for one of the possible medications to prevent the proteases from getting to the HA.
Yeah, so some anti-influenza, the problem is they're all cellular proteases, and usually those cellular proteases have some other role as well. So that's a bit of a challenge to be able to use that specifically. When we talk about HIV, there's an HIV protease, which is a specific viral protease, so that's a much better target. And also true for some of the flaviviruses, which have very specific kinds of proteases, and so if you can find a way to inhibit those, that makes more sense.
You know what might kill it? If we just flood the cell with hydrofluoric acid. That might do it.
Kill it. 5. Quick, vote. Oh, which reminds me, there's still a few people who haven't registered their clickers. If you're not seeing clicker points, then make sure that you email me your clicker number, and I will make sure that those get added to your name. I'm getting all of these people, there are like 3 or 4 people who are clicking away and getting lots of points, but I don't know who you are. So if you don't tell me who you are, then you're not going to get those points. So how many do they have? 8. There are some orthomyxoviruses that have 6, but all of the A's have 8. Okay, so how do we get this messenger RNA production? Why do these viruses decide that's the way to go? Now, you've got to get into the nucleus to steal all of the caps. So this is that whole process here. You have cellular pre-messenger RNA. Now, why pre-messenger RNA? We all remember back last term when we were taking molecular biology, or the last time that you took molecular biology, that capping, or you've forgotten it, as the case may be, the capping process happens co-transcriptionally. So as soon as the RNA polymerase gets past that promoter and starts to make the messenger RNA, the capping enzymes come in and make the cap. So well before that messenger RNA has completed, you've got these cap structures, which are there. And to totally over-anthropomorphize, the orthomyxoviruses take advantage of this and have a cap binding protein, which is PB2, and PB1, which is an endonuclease that will chop off this cap at an adenine residue. That adenine residue can now base pair with the 3' end of each of the genome segments. So what does that mean about each of these genome segments? What's the 3' nucleotide of every single one of these? U. So even if you don't know the rest of the sequence, you know 8 bases of all orthomyxovirus genomes, because the first nucleotide is always going to be a U, because the endonuclease is always going to cut right after an A. Once that's done, now we have a primer with a 3' OH, a template, which is each of these genomic segments, and these RNA-dependent RNA polymerases, whose structures are very similar to the DNA polymerases. I don't know if anyone remembers back to polio, whenever that was, ages and ages ago, which will still be on the midterm. The structures of the RNA-dependent RNA polymerases are all DNA polymerase-like. So they need primers. So where do those primers come from? In this case, it's the primer which is coming from the cap, which has just been stolen for mRNA production. For genome production, it turns out that they can replicate without these caps and without the primers. How that works is not entirely clear. So, but at least for messenger RNA production, we've stolen a cap. We extend our way down each of these genome segments. We hit a stretch of U's, but it's not hundreds of U residues. So what happens to that RNA polymerase? It stutters. Have we heard about stuttering before? Yes. It's where the polymerase sits there and spits out lots of what nucleotides? A's. So you have a poly A tail. Woohoo! So now you have a messenger RNA. So steal the cap, use that as the primer to make the messenger RNA, and then stuttering at the end of the genome. Yeah?
So PB1 subunit has both endonuclease and
Polymerase activity, yes. And that's it's back in the table that we looked at before. So that's there. You might ask, what is PB2 doing? Good question. Yeah, it helps to bind the cap. What about the PA protein? Maybe helping to bind, maybe not. So I would consider those proteins. PB2 is definitely cap binding. PB1 has this endonuclease and extension activity. PA is there. Quite exactly what it's doing, nobody really has a good handle on it. Almost all of these messenger RNAs which are made do not get spliced. They just get translated directly. But if you remember way back when we talked about the cartoon, how many proteins are made from these genomes? Like 10. So we've got 8 genome segments. How do you make 10 proteins from 8 genome segments? You have 2 of them that get spliced. So 2 of the messenger RNAs get spliced. But the vast majority of them don't, vast majority being 6 out of 8. Those get spliced, but not always spliced. And it turns out that's alternative splicing, which will give you the, the matrix proteins and the NS proteins. So this is how you get a messenger RNA. How do we get genome? It's all about the NP protein. So the NP protein, if you have enough of this extra NP protein, which means, yes, we've replicated our genomes, we've translated all of our proteins, we've gotten the import of those NP proteins, we've made genomes, they've bound now to NP. If you have still extra NP around, that says, hey, it's time to make genome as opposed to make messenger RNAs. So if you have NP around, that changes the activity. And so now, instead of stealing a cap, PB1 can actually start with ATP, again, binding to the U, which is at the 3' end of every single one of the genome segments, and then replicate its way down to the opposite end of the genome. And when, again, you have NP protein around, you don't stutter. Because if you were to stutter, you'd end up with longer and longer genomes. And that might work for a couple of genome replication rounds, but you'd end up probably blasting open your provirion. So when you don't have stuttering, that's when you have extra NP around. If you don't have extra NP around, that means you haven't made the messenger RNAs and translated all of these things. So you need to be making more messenger RNAs. So it's all about the concentration of this NP protein. The only ones we haven't talked about so far, so we talked about hemagglutinin, neuraminidase, PA, PB1, PB2, NP. What do we have left? M1, M2, and NS1 and 2. So NS2, that's the one which is still associated with the virion. It seems to be involved in getting those whole genome segments together with the NP, et cetera, out of the nucleus. So PA, PB1, PB2, and NP all have nuclear import signals, but the nuclear export signals are only on NS2. So NS2 has to associate with those genome segments to get the whole thing back out. The other one is NS1. So what does NS1 do? NS1 is involved in host regulation, and NS1 does all kinds of fun things regarding transcription. So we talked about capping, which is being done co-transcriptionally. As soon as the RNA polymerase starts, you have the capping enzymes come, bind to the cap, make the cap. But then what happens at the 3' end of your messenger RNA normally? You get a poly A tail. But how do you make a poly A tail normally? So you have cleavage and polyadenylation. And cleavage and polyadenylation are stimulated by cleavage and polyadenylation stimulation factor. So CPSF. So the CPSF will give you poly A tails on your messenger RNAs, the cellular messenger RNAs. And the PABP is what? Poly A binding protein, which will stabilize those tails and get all of the cellular messenger RNAs out into the cytoplasm where they get translated and made into proteins. Well, again, to totally over-anthropomorphize here, if you're a flu virus, do you want the cell to be making all of its messenger RNAs and translating them? Heck no. You want to be making all of yours. And so by blocking the cleavage and polyadenylation specificity factor, there are going to be very few messenger RNAs that make it out into the cytoplasm, the cellular messenger RNAs, and at the same time, polymerases will get to these cleavage and polyadenylation specificity factor sites, and you'll end up with a lot of unprocessed RNAs that are present in the nucleus, which is great because it means a ton of caps that the influenza virus proteins can steal or snatch and put onto their genomes. Yeah?
Doesn't this kind of go against the idea of a successful virus not destroying the host in order to replicate? I mean, this is kind of like anything that the cell needs to make in order to stay sufficient is now potentially being blocked.
Yeah, so again, over-anthropomorphizing here, but from the virus's point of view, it's really stupid to be killing off all your cells, right? So what seems to happen here is that there's also pretty decent defense mechanisms against flu, and not all of the cells die in this process. And probably what's happening is you have a few cells that are given up at this point, but there's still plenty of other cells that actually can get infected. But this also brings up the whole issue of what's your reservoir species? How are all of these things working? And the answer is, if we get to it, which we may or might not to, reservoir species are probably avians, so probably birds are the place where almost all of these are replicating. And flu is, as we'll see, a pretty self-limiting virus in humans. And most people actually don't get that sick from it. It's just the few that do, which is still an appreciable percentage because so many people get infected, that it seems to be able to maintain itself. Yeah?
Once there's enough NP protein, do they stop blocking the CPSF and the PABP?
Not sure I quite understand your question. So it basically is the level of NP protein determine also the level of NS1 and NS2? Maybe that's another way of asking your question. Basically, what happens is you end up switching over from, again, making these messenger RNAs, which will then get translated, to making genomes. And so if you're making genomes, then you're not going to be making new messenger RNAs for NS1 and NS2.
So can the cell keep on making their own messenger RNAs?
Well, again, there will be some production of these other messenger RNAs because there still are some things that, again, you need an RNA polymerase to be making these caps and these things. So it's not a complete shutdown. It's inhibiting those. So it's not as extreme. If you look at a influenza-infected cell, you'll see reductions in transcript. You'll see reductions in translation. But it's not as complete a reduction as you see in something like polio, which is cleaving, just getting rid of all of the translation factors which are required for getting translation. So you will still see some production there, and it's just slowing them down, but it's not completely wiping it out. The other thing that happens is blocking PKR. What the heck is PKR? We talked about PKR yet? I don't think so. So this is a protein kinase that's stimulated by double-stranded RNA. And that PKR actually also leads back to these eukaryotic initiation factors, so translation initiation factors. PKR will phosphorylate EIF2 to block it being recycled and going back in to make more new proteins. So PKR is a virus defense mechanism that the cells have that say, hey, I've got double-stranded RNA around. If I got double-stranded RNA around, that probably means there's an RNA virus that's just trying to replicate in my cell. This is a bad idea. And if it's replicating its genome and trying to make messenger RNA, it will be good to block translation until we can get rid of it somehow. And the viruses know this, or have figured it out, evolutionarily speaking, and then we'll also try and block that activity. So their genes still will be translated and be made into protein. Yeah?
So it shuts down all translation. It's just a blanket translation.
It is a blanket translational stop, per se, but again, these are not complete kinds of things. And the viral proteins are dependent on exactly the same kind of initiation factors. So the idea here is that PKR would block all of much of the translation to try and block the translation of the viral proteins. And the viral proteins have evolved to block the activity of PKR. But if you do have PKR induction, and maybe, so we have, again, after the midterm, who cares about after the midterm? We'll have a couple of guest lecturers. One of them may actually talk about PKR in terms of virus defense mechanisms and innate immunity, that's part of that process. Okay, so we've shut down cellular transcription. We've stimulated translation to avoid the cellular mechanisms. Now we need to get out. As I mentioned before, we looked at the micrographs right at the beginning. Budding happens at the plasma membrane. So you have all of your nucleocapsids, which are here, that then get associated with the HA and the NA, as well as your M2 protein, which is this ion channel. And these then bud off of the host membrane. The neuraminidase is probably cleaving off the sialic acid that the virus would normally bind to and allow genome release to take place. One of the big questions about how these virions get put together is how you end up with 8 and only 8 of these genome segments in each of the virions, and only 1 of each. Because if you think about it from a just statistical point of view, if it's a random mixture of each of these 8, and you need all 8 of them in order to be able to replicate, it's highly unlikely that you would get all 8 and only 1 of each into each virion. And that's still a very open question. We don't know how that works. And it could actually be really, really useful in terms of understanding some of the viral disease that we'll talk about in just a second here. Okay, nice overview, again, from the Flint textbook that I will try and remember to take over today. So it should be hopefully on reserve for next week if you're looking for it on Monday. At the back of that textbook is probably the best part of it because they have overviews of all the different virus replication schemes there. This is the one for influenza, binding again by HA to sialic acid, comes into the endosomes, pH changes, the nucleocapsids are released, they go into the nucleus, they get made into antigenome, which is now positive strand RNA. That then gets made into negative strand. How does that happen? It's through the translation of all of these individual segments, which have stolen caps from the host RNAs. Those get exported, then re-imported back into the nucleus to give you production of your genomes. These genomes get associated with the NP protein. Once you have all of that and NS1, that allows you to export into the cytoplasm. These guys end up at the membrane together with Na and HA, which are being translated at the endoplasmic reticulum, being modified. Proteolysis takes place. These guys end up at the membrane. You have budding, and the whole piece comes off. So it's a nice review of the whole process. Yeah?
For the replicase complex and nucleocapsid complex, are those the only proteins that we know of that are bound with them from their transport, or with each genome segment, from its transport from the nucleus to the plasma membrane where it buds?
So is it just NP and polymerase associated with nucleocapsids? No, you also have to have that nuclear export protein that associates with them. So you also have to have NS1, which associates here with your nucleocapsids in order to get everything out.
But all the genomes they have the identical proteins associated with them, each segment does.
Yeah, each segment has PA, PB1, PB2, and NP. So there doesn't seem to be any differentiation between any of those. Yeah, it's interesting. So yeah, no sequence-specific kinds of things that are happening except at the very 5' and 3' ends, which is where you have the base pairing that takes place. And that actually seems to be mostly binding to the double-stranded RNA, which is there, rather than the base pair specifically. Okay, more questions on the molecular aspect. You don't have any? I've got one. Influenza protein most important for membrane fusion is alphabet soup, HANA, PB1, PB2, or NS2.
As they make their anti-genome, the cell doesn't attack the double-stranded RNA?
Those levels don't seem to change a huge amount. No, they're certainly made from each of the replications where you make your messenger RNA. But I don't think that the concentrations go up particularly, that they get transcribed and translated specifically and preferentially. I don't remember that there's a big difference in terms of those.
So they're just basically, those antigenomes are just in danger until there's a status 2.
You guys are still messing with me, but I want 100%. This is what we're trying for. And yes, it is HA. No, I'm not trying to trick you on any of these things. At least I don't think I'm trying to trick you. It's what I'm thinking. So now let's talk about disease. All this nasty stuff here. So basically, symptoms of flu. This is according to the Center for Disease Control website. You can read through all of these if you like. I think the main one down here, if you really can't get out of bed, it's probably flu. If you can get out of bed, it's probably not flu. So that's the sort of the main thing here. The only other one I wanted to mention is that vomiting can occur. A lot of people talk about the stomach flu. I usually vomit when people talk about the stomach flu because it's not the flu. So it's very, very, very rare among people of our age that influenza will lead to any kind of GI reaction. This is really about a respiratory disease. So, okay, fine, you feel bad. But
so if you do have a stomach issue,
mm-hmm.
what virus would it be more likely to be if it was a virus?
So many of the enteric viruses that we've talked about, so hopefully it's not polio, but Coxsackie virus, there are many of these other just general enteroviruses. And again, polio is an enterovirus, one of these things. But the main disease phenotype, of course, has once it gets out of that part. But yeah, the flu, the stomach flu is really, I'm not quite sure where that came from, that whole business. But the big problem with flu being a respiratory disease is that also, as you remember, all of the virions are being produced by these cells, which are in lung epithelia. So what happens? You sneeze. And so that's where you get massive spread of these diseases and the aerosols. And one of the things that I always like to say is aerosols are really good at getting about about a meter away from you, which means that if any of your seat is next to you, that means that they would be exposed to you and your flu viruses, which are there. And what that means is actually it's a very high infection rate in any given flu season. Most people who actually don't have really nasty symptoms, and the symptoms can vary really massively. So if you just think about the US, and these are the numbers here for the US, 5 to 20% infection rate in the US in any given season, that's up to 60 million people. That's a very, very, very large number. And of those, most people don't get really sick. But again, if you've got these really large numbers, even if a small percentage of people end up getting sick, you'll have lots of hospital visits. So it's hundreds of thousands of hospital visits in any given year are due to an influenza infection. And on the order of 30,000 deaths per year. Now, how many deaths were there in the US from Ebola ever? One. And we got 30,000 plus deaths per year from flu. And what do we see on the cover of things?
Yeah, but that's Ebola. That's scary.
Yeah, exactly. Flu should be scary. So that's, maybe I'm... It needs a new name. There we go, yes. And don't call it stomach flu. Don't call it stomach flu. Now, there you go, exactly. Or bird flu. Bird flu got a few more people as well. Swine flu also gets people as well. So now, that being said, of the large number of deaths that happen in any given year, most of those, the vast majority, are in people who have other kinds of symptoms, usually those who are older. And so the vast majority of these deaths are with extra conditions that are involved with that. And we'll take a look at that a little bit later on. Now, everything from here on up is all US. Worldwide, however, there's still lots and lots and lots of people who die in any given year from flu. So it's definitely a big problem.
You say 98% of the deaths?
Of the deaths are in people who are above 65 in usual years. We'll get back to that. I know exactly what you're thinking about, Sarah. So we'll get there in just a second. So one of the things, because there actually is, or should they, are lots of flu deaths in any given year, the CDC, unlike the rest of the population, does do a really good job of monitoring all of the flu outbreaks. So these are the data up until, I think, last week of flu this year. So most of the measurements here, these are thousands of specimens that have been sent in. These are from special reference labs that are spread all throughout the country. When someone comes in, the hospitals will then take some of these samples, send them off to the CDC, and they will get measured because they think that there may be some flu which is circulating there. One of the things they do is they check to see if it's A or B. A here, this particular year, this varies a little bit from year to year, peaked a little bit after the New Year. This is just the number of weeks here. So New Year's is right here. This is week 2 of the year. Peaking here around week 8. So end of February, and now we're coming down. One of the interesting things this year, which was actually kind of strange, is Influenza B, which also causes disease, usually not as severe, was much later. And so there's usually in any particular year you'll have peaks that happen at just about the same time. Of the samples that got sent into the CDC, between 10 and 20% of them were actually positive. That means that 80% were negative. So there was still quite a few samples that got sent in that actually weren't there. One of the things that's important, and we'll talk about this when we get to the vaccines in just a second here, is trying to figure out what kind of flu it is, which is under the infection process, because that's what has to do with the vaccines that we'll have in a previous year and then also are using for the following year. So testing for these particular types, and most of you heard about H5N1, H1N1, H3N3. What do the H's and N's stand for? Hemagglutinin neuraminidase. Exactly. So there are, I think, 24 different hemagglutinins and something like 18 different neuraminases that have been reported so far. But the vast majority of human disease is coming from H1N1 and H3N2. And this year, it was almost all H3N2. In previous years, there's been more H1N1, and this TDM09 is probably what Sarah wants me to talk about, which is the swine flu from 09, which actually had much higher mortality in younger age groups here. So we'll get back and talk about that. But there are still a few other ones here. This is an H3N2, which is slightly different, but the vast majority H3N2 of this year. It turns out that this year, the H3N2 was not as well protected by the vaccine. The H1N1 was much better protected by the vaccine. And again, later B forms that have happened here. So this is really trying to look at breaking down all of these things. Thousands of specimens that were brought into the CDC. Again, the vast majority are these H3, and almost all of those are this particular M3C, which was not, again, particularly well protected against the vaccine. The H1 and TDM09, almost all of the cases here were in fact this same one, which is still circulating again from the 09 outbreaks. And interestingly enough, the diversity in the B's is very, very low. So H1 very low, B is very low, A a little bit more diversity. Yeah. So in these seasonal flus, B's usually lower mortality and A's higher mortality. And also shows the severity of the symptoms. Same kind of thing here. So yes, vast majority of diseases are happening for older people, but this is why, as a parent, I'm very concerned that my kids get vaccinated and everyone around them gets vaccinated, because there are, in any given year, around 100 pediatric deaths. And pediatric here, by CDC definition, is under 5. So in any given year, there are about 100. This year was actually a little bit lower relative to some of the previous years in terms of kids dying of influenza-like disease. So there certainly are numbers here. And again, hate to be beating on the dead horse here, but how many died from this? And how many are dying from flu in any given year? So this is just another way of looking at that. This is the 65-plus year group who are, in fact, hospitalized, and those, unfortunately, hospitalization is also directly connected with dying. That's where people go. That's where people die, is in hospitals. But that's because they have really nasty symptoms. And so 65 and up, and then down here are the 0 to 4s. So there's still a reasonable number, but nothing like this age group right here. As I mentioned before, almost all of the people who come down with disease have some kind of pre-existing condition, which then is clearly contributory to seeing that. The one that I did want to emphasize here is, at least with the children, the majority here actually have no obviously known, well, not the majority, but about 50% have no obviously known condition, whereas for the older, i.e. everyone who's above those ages, usually has some kind of underlying. And for kids, in fact, asthma is one of the major ones here, which seems to be correlated with disease. So again, this year, we're a little later than most years in the past, although the year before it was even later that we had our peak in terms of influenza disease. And then there's this gray line back here. That's the swine flu in 09 and 10. That was much, much, much earlier. So 52 is the end of the year. 10 weeks earlier. So this particular flu started really in October. So when should you be getting your vaccine? As early as possible, September-ish. They have this wonderful, good way of looking at where flu is in the U.S. All this FluView stuff is at the CDC. You can see where flu is circulating, where it's gone down to sporadic. There still are some flu cases in Oregon, but the rest of the country is looking like this, and this is literally through week 17 now. So just to finish up, and unfortunately I didn't have a chance to talk more about the vaccines here. Maybe we'll talk about it next week. Well, no, got the exam Friday. We'll see. Last favorite question for today. Everyone should get this one right, I hope. Flu season 2016-17 is early and usually later than usual. Currently above the epidemic threshold. Most individuals find more deadly than usual. Your gut. But it's not flu, it's your gut. Exactly. No. Data. Characteristics. It's Star Trek. very last second. Again, I think people are messing with me. One person. You're not going to do that on Wednesday. So Monday will be a review. Wednesday will be the exam. It's just the stuff we got today that will be on the exam. So all the vaccine stuff.